Did anybody forget a laptop here? Like 2 weeks ago, on the way in the back? No, nobody? All right, so here we're back. Let's continue where we left off. And remember, I also made last year's videos available, right? So you can check those out and double check if I forgot to mention something in the class that is really important, because I really had like more time to cover the things last term. To do a task of copying your entire textbook. You can sit down by yourself and try to copy it by hand, but if you divide the task among at least 30 of your classmates, you can copy it a lot faster. Okay. So the idea here is to divide and conquer. So here is another problem. How do the cells know when a certain piece of DNA was replicated in comparison to a piece that was not replicated? So there are several strategies that the cells use in order to distinguish the DNA that was already replicated from one that was not replicated. You're going to have to know only a couple of them. Okay. So first thing that's going to happen, you're going to have a recognition complex of proteins that recognizes the origin of replication. Okay, so you're going to have certain proteins here that go to the origin of replication and recognize that site, and at that point is when you start to do those interesting things to DNA, like you bring in the proteins that are going to associate with the origin of replication, right? You bring the DNA A protein that associates with the DNA and makes a little loop, meaning it's bending the DNA and it's going to cause a little bit of a bubble to form, right? At that point, you're going to bring another protein that is DNA B that is going to start to open up. It's a helicase. Okay, so DNA B starts to open up that replication site, and then you're going to bring polymerase that starts doing the polymerization. So here the question is, what happens with polymerase recognizing what needs to be replicated? So the first thing that comes into play here is the presence of those proteins that are associated with old DNA. Okay, so the DNA of the template that was not replicated, that is serving actually as a template. So these proteins are called MCM proteins. Okay, they also happen to be helicases. So they associate with the old DNA, and they keep on associating with the old DNA until that DNA gets replicated. Once it gets the replication that it's supposed to undergo, these MCM proteins dissociate, so they're not there anymore. So that is a first sign telling the polymerase machinery that if you do not find MCM proteins, don't replicate the DNA because the job was already done. Okay, so this is one strategy that is quite important. Okay. I didn't even put more strategies there. So that's the one you have to remember. Okay, presence of MCM means replication wasn't done. If MCM is not there, it means that piece has to be replicated. Okay. So another difference that we have here when you compare the replication I was seeing before with prokaryotes and eukaryotes is that when you deal with the prokaryotes, you basically have to remember only 2 polymerases, right? You have polymerase 3 that does most of the job. That's the alien-looking thing that has a tau and 2 arms and has the eyes, right? And does that the whole job of replicating the DNA. Besides polymerase 3, you also have DNA polymerase 1, and that one is important for the lagging strand, because polymerase 1 is what kicks out RNA and puts in DNA, right? So it's going to replace those Okazaki fragments and make linear the DNA. It's going to make the DNA molecule. So 2 polymerases to remember, and also, of course, you need to remember the subunits of DNA polymerase 3, right? Now, when you look at eukaryotes, you have a lot of polymerases. going on over here. So you have like 13, at least 13 different polymerases that are working with the eukaryotes. You don't have to remember or know all of them, so you're going to have to know a few of them. Okay. So important ones to know here is polymerase alpha and polymerase delta. So those are very important polymerases that are involved with nuclear DNA replication. That means the linear DNA that is inside the nucleus. Do you have other genomic DNA in eukaryotic cells? Yes, what is it? Mitochondrial DNA, I heard all the way in the back. Who said that? Yes, mitochondrial DNA. You go like, yeah. Okay, so is that considered, genetic material, mitochondrial DNA? Yes, yes. But it has a different form of replication, right? Mitochondrial DNA is circular, just like bacterial DNA. Remember, mitochondria was that endosymbiotic process of an ancient bacteria that got engulfed by this other, bacteria, and they became, eukaryotes. Okay, so replication of mitochondrial DNA happens in a different way. Okay, when you're talking about the linear DNA inside of the nucleus, you have to know polymerase alpha and beta. Okay, so here, polymerase alpha. You have 4 subunits of this alpha that are going to work as that primase. All right, remember in prokaryotes you have a primase that goes there, falls off, then polymerase comes in. In this case here, you have a piece of your major polymerase alpha that has the primase activity. So this polymerase comes with its own primase. So primase puts in the primer and starts the polymerization, right? Now, polymerase A has the DNA polymerase activity, but is not very good at it. So we say it has low processivity. Low processivity means it has a very short attention span. It starts to do the job, and then it gets distracted, it falls off the DNA, and then the replication wouldn't really happen if you only rely on polymerase alpha. Now, another polymerase comes in and picks up the job. So that is polymerase delta. Okay, so that one has high processivity. That one is the one that comes in with that, ring on the string trick. Okay, with prokaryotes we call that beta clamp. In eukaryotes we call that pCNA. Okay, so the ring that goes on the string on the single-stranded DNA, that's the pCNA. So polymerase delta brings that, and that allows it to have high processivity. Besides that, you always remember polymerases, RNA or DNA polymerases, they're always going to make the polymer that goes 5' to 3', right? So besides that, this polymerase delta here, it has a very important 3' to 5', so backwards, exonuclease activity. So this is very important to correct mistakes that the polymerase could have made in the first place. So important correction machinery here. Now, this subunit has, besides all that, it also has this RNase age part to it. And it works with a polymerase delta, and this RNase age is what does the job of polymerase 1 for the prokaryotes. So this guy in eukaryotes is going to remove the RNA primers, and he's going to replace the RNA primers with the DNA. Okay, so here this is a comparison of properties between the two systems here. Okay, so what happens with the mitochondria? Right, so they have their own system of replicating the DNA. Mitochondria can replicate the DNA, but on its own, it doesn't need the whole cell to be dividing for it to replicate the DNA. And the mitochondria, they're actually very active organelles. So they are going to divide or fuse depending on the demands of a particular cell. Okay, so let's say that you are exercising a lot. You're using your muscles a lot. So when you're exercising a lot, you need a lot of energy production. So your mitochondria in your muscles, they're going to act differently when you compare it to the muscles of a person that is mainly sedentary. Okay, so the mitochondria in your muscles, they're going to start dividing, and that division of the mitochondria, it doesn't really go with muscular cell division. It doesn't go with muscular fiber. So mitochondria does their own thing on its own. It knows when it needs to divide, when it needs to fuse. It's quite dynamic. The important polymerase that is acting in the mitochondrial DNA is this polymerase gamma. Okay? So here is showing this circular, relatively small mitochondrial DNA. And here the polymerase that does the replication is the gamma. Now, the inheritance of mitochondrial DNA is only passed down from the mother to the child. Okay? So when you look at your mitochondrial DNA sequence, you're going to have your mother lineage DNA sequence. Okay? So you don't really see mitochondria from the sperm that gets incorporated when you have fertilization of the egg. And there are several reasons why that happened. We're not going to go into detail on how that is happening. But one reason, keep in mind that when you have that sperm that is swimming around and looking for the egg, right? The upper head here of the sperm mainly contains a bit of fusion proteins at the tip, and it contains the chromosomes that it is carrying. Nothing else here. There are no organelles there. The part that is containing mitochondria is actually here going to the tail of the sperm. So when you have fusion, only the chromosomes here that are going to be dumped inside of the egg, and the rest of the sperm is going to be discarded. So this is like one of the reasons why you don't get mitochondrial DNA or mitochondria from the fathers. Okay. All right. So when you look at overall eukaryotic cells, we also know that plants also have this, besides having mitochondria, plants also have chloroplasts, and those have basically the same type of DNA replication that you see with mitochondria. So you have these two types of replications that are independent of the eukaryotic cell division. Okay. Parts to it. Polymerase alpha is the first one that gets there. The first part that is going to work is going to be the primase. Because remember, the DNA polymerase cannot start synthesizing the polymer if it doesn't have this group of OH that is already there. It can only extend. It cannot start from nothing. Okay? So one enzyme that can start from nothing, those are your RNases, RNA primers. The primase activity. It is RNA polymerase. No, actually, it is an RNA polymerase, but it doesn't make a huge polymer. It makes just a small polymer. So in order not to get confused with other RNA polymerases that we're going to see later, so we call it a primase. That's the job of that RNA polymerase. All right, so here is another difference that you encounter when you're comparing your prokaryotes to eukaryotes, right? Remember, with your prokaryotes, you don't have to deal with that DNA compaction, so you don't have to deal with the histones, right? So here with the eukaryotes, you have to deal with the histones, right? So remember, histones are basic proteins. They have lysines and positive charge, and in order to access the DNA for polymerization, you need to take out those associated proteins. Otherwise, you cannot really open up the DNA. How do you take out the proteins? You have to modify them. you have to cancel that positive charge, right? So you have those writers, enzymes, that go there and put acetyl groups on the tails of your histones, and that neutralizes the charges of those histones, and then those are going to fall off. And there is a lot more for that process, but of course we're sticking only to the basics here. So acetylation of those histones are going to make them fall off. Then after the DNA is already polymerized and you have to make it compact again, then you're going to modify the histones again, and you're going to take out those acetyl groups to make the histones associate back with the DNA. So at the beginning of polymerization, you have acetylation, and then after polymerization, you're going to have deacetylation. So what you do here, you think, what is it that you need depending on the stage of DNA replication? If you need to kick those charges out, what do you do to the histone? What do you do to the positive charge? If you need to put them back in, what is it that you need to do? Okay, make sure that you understand the sequence of events in your mind. And we already talked about those epigenetic patterns and the histone code, right, before. So here's just to emphasize that when you are replicating the DNA, you're going to have double the amount of the DNA that you started with. That means you're also going to need double of the amount of histones that you started with, because if you do not make more histones, you're going to end up with not enough proteins to pack up your DNA at the end. So as soon as you start your DNA replication, you also start synthesis of histone. Okay? So you start histone synthesis at the same time that you start your DNA replication. And remember, the histone code is very important for each differentiated cell, because the daughter cells have to know which genes to keep active and which genes to suppress, right? So that is written on this histone code, right? One clear example of the importance of histone coding, right? This is epigenetic factor, right? One very important example here is seen with dosage compensation when you're talking about X chromosome inactivation. So what does that mean? Well, if you are born a woman, you come with 2 X of your sex chromosomes, right? One from mom and one from dad. If you are born a man, you come with 1 X and 1 Y chromosome. So if you look at the possible number of genes to be expressed in the male from the X chromosome, you're going to see that it is half of the amount of genes that you have with the cells that are from a female. So that will be a problem, because if females would express twice the amount of genes that is present in the X chromosome, we wouldn't really have a very stable life compatibility. There will be a lot of problems associated with double of the expression of these genes that are in the X chromosome. Okay, so what happens here is that one of the X chromosomes in all of the somatic cells of females, one of the X chromosomes gets inactivated. Now how? Well, you're going to have special histones that are heavily methylated. Remember, methylation of histones stabilize the positive charge on the histone. If you stabilize the positive charge, that histone is really going to be stuck to the DNA. That is going to inactivate genes. So if you look at the X chromosome, at the extra one here, that you don't want to express, you're going to have a lot of those methylated histones, and you're going to have a lot of, like, this whole chromosome is going to be inactivated. Okay? So this is what you call for dosage compensation, and is solely dependent on epigenetic factors of histone coding. Okay, and it's quite interesting that sometimes you have problems during gamete formation of the sperm. Sometimes you get a sperm that have X and Y chromosome, and then it fuses to the egg that already has an X chromosome, and then you end up with an egg that has 2X and a Y. Anybody took genetics? Yeah. So you remember this kind of problem? Yeah. And then what happens to these cells? Is this going to be a male or a female? It's going to be a male. If you have a Y, it's going to be a male. Now, what's going to happen now with the X chromosomes? One of them is going to get deactivated, just like in the females. Okay? So this is like, it's amazing that this type of histone coding is passed on even when you have some type of genetic problems. Okay, so this is called dosage compensation. All right. Okay, so here are some more terms that I had not mentioned before, and also some other problems that you're going to encounter with your eukaryotic chromosomes. So here, when you look at your eukaryotic replication, remember it's linear. When you compare it with your prokaryotic replication that is circular, so now we run into this other problem, that is the presence of the telomeres. So telomeres at the end of the linear chromosomes. Okay. So here, let's see how we're going to solve that problem. So several problems related to telomeres. Now, I put here again this circular DNA, showing that, again, with prokaryotes, you have one origin of replication and one termination of replication sequence, right? Now, this whole thing here, this whole circular DNA, is called a replicon, okay? So from origin to termination, that's the replication, from origin to termination, you have this replicon. And then again, when you look at eukaryotes, you have many, many, many replicons, right? Because you have many, many origins of replication. So now looking here at your linear DNA, okay, again, you're going to have several start points, and you're going to have issues that when you finish the replication of the linear DNA, you're going to get to the end of that stretched line, and you're going to have a problem of finishing up the replication with your lagging strand, because you are not going to have a free 3' OH at the end of your lagging strand that you can synthesize your missing part of DNA over there. Okay. And why is this a problem only for the lagging strand? Well, because when you look at your leading strand, it is continuously synthesized, and by the time it finishes the DNA synthesis, it finished already the whole thing. So it's done its job. Okay. So here with the lagging strand, we're going to have a problem because of those primers, because of those Okazaki fragments. So that is really a big problem for your linear DNA, because every time you do cell division, theoretically, the chromosomes are going to shorten by a little piece. Okay? So if your chromosomes shorten too much, you're going to hit important genes on those chromosomes, and you're going to cause cell death. Okay? So that those hanging ends are really big problems for your linear DNA. Okay? In order to solve that problem of those telomeres, you have this enzyme that is called telomerase. Okay? So telomerase is a very ingenious way that the cells figure out on how not to shorten those linear chromosomes every time you have cell division. So how is this that telomerase is doing this job? So telomerase is made up of proteins and also it has a piece of RNA on its body. Okay? So this enzyme uses a piece of RNA in order to associate with that free hanging piece of DNA that is missing the double helix. So this is a single strand of DNA. Okay? What is it telomerase is going to do? Telomerase is going to fill that end and say, you know, here is a sequence of nucleotides that I can recognize because I have a piece of RNA in my body that can do base pairing with those nucleotides. By the way, every species has a different sequence of this template of RNA that is present on telomerase. Okay? So for us humans, there is a specific sequence that goes here to this telomere, to this telomere side, interacts with it, and then what telomerase does, it starts to make DNA out of nowhere, and it's using its body as the template to make new DNA on this lagging strand right there. So it extends the DNA that was hanging out of that telomere. Okay, so you're extending it. You had a piece here, you couldn't really synthesize DNA, so this guy goes there and stretches it out, makes new DNA over here. Okay? So technically speaking, telomerase is a reverse transcriptase. That's the name of the enzyme that uses RNA to make DNA. Okay, have you heard of reverse transcriptases somewhere else? Like HIV, right? HIV has reverse transcriptase, or some viruses, they can do reverse transcription from RNA material to DNA, right? So some viruses have those types of proteins as well. Okay. So in our cells, we have this telomerase that does that job. Now, interestingly, this enzyme is not very active in your cells that are differentiated. Okay. There are certain cells in your body in which you have active telomerase enzyme. Okay. And this is a bit of a correction, or a bit of a different talk that I had from my previous lectures. Okay. Because I learned when I took cell bio, don't ask me when. When I took cell bio, I learned, well, telomerase is only active in gametes, in embryos, and in stem cells, and that's it. And I never stopped to think if that would make sense. This is the thing about this class, right? It's so much material, it's so much thrown at you at once, that a lot of times you're basically concerned of memorizing it and eating it and regurgitating at the test and passing the class, and that's it, and you never have to look at it again. Oh, sometimes you do. Okay, so then what happened? When I was looking through this lecture, I'm like, wait a minute, it doesn't make sense. Why doesn't it make sense? Well, because you have certain areas in your body that you have constant cell division and turnover. One place is in your gut, in your intestines. You have intestinal shedding all the time. You keep on replenishing those intestinal cells, the lining this one cell line in your intestines. Now, if you do not have active polymerase in those cells, you're going to run into trouble, because continuous cell division is going to shorten those telomeres, and it's going to damage those cells, and they're going to die. Okay, so you have a problem there. So this basal level of telomerase in those very highly dividing cells, it's a bit higher than in other differentiated cells in your body, in which you don't really see high turnover or high cell division. Okay. So besides your gametes and your embryos and your stem cells, you do have certain cells that have active polymerase. But those here are really the most important cells with the most active telomerases, because those really undergo a lot of cell division. Okay? All right, so here's with the telomerase. Okay, So what is happening here with the sequence of DNA that telomerase is adding, right? That sequence is repeat sequence. And again, you have different repeat sequence for different species, right? And it extends so that you are not going to shorten that chromosome too much, and you're not going to hit important genes to to damage the cell function. Okay, so again, when you look here at the end of your linear chromosomes, you know that another problem that it poses, besides the problem of DNA replication, another problem here is that if you have linear stretched out DNA, and you have several pieces of linear stretched out DNA, you can have base pairing of those flying DNA molecules. They can just interact, and remember, these telomeres all have repeating sequence. So they would be very prone to just base pair with the telomere of some other chromosome. And then you would have chromosome fusion, and that's a big problem. That's a big problem for the cells, okay? So in order for that not to happen, you have certain proteins that are going to bind to those repeating sequences, and they are going to shield the DNA and prevent it from fusing to other telomeres. Okay, so you have protein interaction that are going to bind there and make those free and not so free. So you're going to stop protein fusion. Another thing that is very important for the presence of those proteins is that they're going to shield the DNA from attack from enzymes that can go there at the end of the DNA and start to chop it up. So those are called exonucleases, right? They go to the end of the DNA, they start to chop it up like Pac-Man, you know, pop, 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 pop. So you don't want that to happen. So protecting by putting a clump of proteins at the telomeres is going to prevent the type of DNA degradation. So there is another trick that the linear chromosomes have to avoid fusing and avoid degradation and to protect the ends of the chromosomes. Okay? So not only you have those protein interactions, but you also have this cool little loop that forms at the very end of the chromosome. So look what happens here. The DNA double helix is going to fold, is going to loop around, and a piece of that free hanging, lagging strand, that piece that couldn't really be replicated, what it's going to do is going to interact with matching base pairs that are part of the double helix. Okay, so this piece is going to be inserted. So what's going to happen here is that you're going to make this double helix out of interaction with a piece of the DNA that is already part of another double helix. So you end up here with a triple helix piece of DNA. So that's a really cool trick. Now what this does is that it makes the end here of the DNA, it makes it look like it is circular DNA. So this is another level of protection against exonucleases. Okay, so you have proteins interacting, and you also have this T loop forming. I always thought that this should be called a loop, but no, it's called the T loop because it's at the telomere. Okay. One characteristic of this sequence is that it is very rich in guanines. Okay, in guanine nucleotides right there. And a reason for that, remember, if you have CG pairing, you're going to have more hydrogen bonds, higher melting point, than if you compare to high content of AT pairing. Okay? So this is going to be a strong hydrogen bonding at this site of 3 helix formation. Bless you. Okay. Okay, so everybody cool with the structure of this T loop? Yeah. Okay. All right. Yeah, you got a question? No, everybody cool? All right. So here, a bit more about the problem of shortening telomeres. Okay. As cells divide throughout, life, again, the somatic cells don't really have a lot of activity of this telomerase, right? So you end up with some chromosome shortening eventually, and as you age, you have more and more chromosome shortening, and that actually serves as a cell clock. Okay. Now, is that related, or does it lead to human aging? Well, I don't really know the mechanisms behind that, but we know that the shortening of telomeres, like by themselves, only the shortening by itself, it's not causative of accelerated aging, but it has to do with the ongoing process of aging. Okay? And we have here, I had put this picture here of two kids with Clifford progeria. I forgot that name. So just progeria is the case of people that have a defective protein that it's not actually telomerase. The protein that is defective here has to do with nuclear lamina, so proteins that are shaping the nucleus from the inside. Okay, so a defect or a truncation, cutting out of the piece of one of those proteins, which is like lamin A, causes accelerated aging. And these kids, they really age so quickly, like for every 1 year, they age biologically by 10 years. Have you seen cases of progeria? Who have seen progeria before? Nobody? One person. Huh? I can see. Oh yeah, in progeria cases, when these kids are inheriting this mutation in lamin A protein, for every year of life, they age by 10 years. So when these kids are 1 year old, they are biologically 10 years old. Okay? When they are 2, so they are 20. By the time they are 10 years old, they are biologically 100. So those kids here, they're like 8 or 9 or close to 10 years old. They are biologically, close to 100 years old. How long do they live? Not long. Okay? Now, think about it. Is this syndrome genetically inherited? All right, let's have a vote here. What does that mean? Can it be inherited genetically, or is it a de novo mutation? Okay, how many people think it is inherited? Okay, how many people think it's a weird freak mutation? Okay, we are divided in the class. When you say something is inherited, it means that you are getting it from either mom or dad, or both, right? When you have this mutation, you're going to age 10 years for every 1 year of life. Are you ever going to hit puberty? Well, by the time they are 2 years old, they would have hit puberty and already be adults, biologically speaking. Are they going to have kids? Hopefully not. That would be scary. Yeah. It's not recessive. Once you have a defect on that lamin A protein, that's dominant. Yeah. So you cannot inherit this from a parent. The only way that this happens is during embryonic development. You have mutation of the gene, and that's just a freak mutation, de novo mutation. Yeah.
Wait, is this only speed up somatic aging or also
So it speeds up everything. Everything in the cell, everything in the body. Everything. By the time they're 1.5, they would be 15 years old, biologically speaking, and yeah, they hit puberty then.
Oh, I thought it was like somatic aging.
No, everything in the body. Yeah. So yeah, it's kind of sad. All right, so this was a spiel on progeria. Okay. So here, mechanism that is being used by telomerase to stretch out those telomeres is here, straight from your book. Okay, here is straight from your book, your T-loop. And here finally comes your first question. So let's get your iClickers. All right, it is running. Why are telomeres useful?
Or it's kind of the same thing.
No. Think in the whole context. Oh, I got 50% of the class divided between the correct answer and the wrong answer. Talk to your friends. Oh no, now more people are giving me the wrong answer. Okay, talk to your friends in the right direction. Telomeres, not the telomerase. Why are telomeres useful? I'll give you a few more seconds to give me the right answer. All right, I'll give up. All right, people, I'll give up here. Let me stop. What is your answer? It is A. Okay, why is it A and not C? Look at this. This is asking you about telomeres. Why are telomeres? Telomere is the end of the chromosome. Okay? Now, A says without them, the chromosomes would fuse together. That's a big problem, right? So the telomeres and the telomere loop and everything is going to prevent your chromosome fusing. Now, they help lengthen the chromosomes? No, that's the job of the telomerase. They stop chromosomes from getting very short. But the telomeres are not doing anything. They're just sitting there. Telomerase is going to stop the chromosomes from getting very short. Okay? And don't panic. I usually do not put this type of tricky questions in the exams. So this is just to exercise your reasoning power. Okay? And if I put the question is going to be exactly the same question, so you're going to get it right. All right. So here, a bit on function of nucleases. Okay. Nucleases are proteins. Okay, those are enzymes. Okay, that they are capable of cutting the DNA or RNA, okay, at specific sequences. So the nucleases that cut DNA, those are called deoxyribonucleases. The nucleases that cut the RNA are called ribonucleases. Then the ones that cut at the ends, they're called exonucleases. The ones that cut in the middle are called endonucleases. Okay? Now, what these proteins do, they are going to recognize a specific DNA sequence or RNA sequence. They're going to recognize specific nucleotide sequences that, and then they're going to grab the nucleotide like a clamp, goes there and grabs that sequence of nucleotides, and these enzymes will cut the backbones of those nucleotides and cut the piece of the DNA out. So those are very important for defense of cells in case they get invaded by foreign DNA or foreign RNA. So two things are implied in this mechanism. The first thing is that the sequence of that RNA or the DNA that these enzymes recognize, that sequence is not present in the genome of the cell that is expressing those proteins. Okay? So what does that tell you? It tells you that if the genome of the cell has the sequence, these proteins would be considered suicidal because they would recognize the sequence in the genome and chop it off and kill the cell. So it doesn't make sense for the cell to produce these proteins to kill itself. Okay? So this sequence is not present in the genome of the cells that are expressing those proteins. Okay? This information is very useful when you look at bacterial cells, because that enables us to manipulate genes with ease. That means you look at the genome of certain bacteria and you say, well, if I want to clone a gene in there, I can use these endonucleases and cut a piece of the gene and paste it to the bacterial gene, either like usually plasmids, right? And then I can express my protein. This is a very, very useful genetic engineering tool. Those are very useful tools. Okay? So it's very important for cloning, right? So here's a little bit of story on this nuclease activities. Yeah?
Not make its own.
Yeah. You mean the gene that is making the protein? Oh, okay. I said a lot of stuff that's sometimes a little bit to digest. So let me go a little bit slower here, but not too slow. All right, so let's think here. You have bacterium right there. So here is the circular DNA. And here, let's put some little plasmids, okay? And then here, let's put the DNA of a virus that invaded the cell and dumped the DNA in there. So there was a bacteriophage, you know, that spaceship-looking, octopus-looking bacteriophage that inserted the DNA in the bacteria. Now bacteria is infected. That bacterium is going to express this protein that is a nuclease. So here, let's put nuclease right here. So this is a nuclease right here. This nuclease is always looking around for a piece of DNA that it can recognize. It looks around, and then it finds that piece of DNA in this invading DNA right there, and it goes there and it chops it off. So that invading DNA, it's not going to allow, you know, viruses to replicate inside of the cell. So this is a defense mechanism. Okay. Let's think what would happen. What would happen if the sequence of DNA recognized by this protein, if that sequence could be found in the genome of the bacteria that is expressing it? Well, that's a big problem because the protein would go there and would chop up this DNA and it would kill the cell. That's what I meant by it's not going to recognize sequences in the bacterium's own genome. Okay? So actually what you call those enzymes here, you call them zero cutters. Because you're going to cut zero of the sequence of the genomic DNA of that bacterium. Okay? So just to keep in mind how mechanistically these things are working, because they have to make sense, right? You want to chop up invading nucleotide polymers, but you do not want to chop up your own. Okay? Those are zero cutters. So that brings us to this idea that you can cut pieces out of DNA and then, either degrade the DNA, or you can cut some pieces out and repair some damage that was done, okay? And this is directly linked with the function of DNA polymerases to correct their own mistakes, right? So here you have this slide that says DNA polymerase has proofreading, okay? And what that means is that when this guy here, DNA polymerase, let's say this guy is DNA polymerase 3. Okay. So DNA polymerase 3, what is it doing? It is adding nucleotides to the, what end is hanging over there? 3 prime is going 5 prime through 3 prime, 5 prime through 3 prime. And then it's going to come to a point in which it's going to make a mistake. It's not very often, but it's going to make a mistake. Okay? So DNA polymerase is adding 5 prime to 3 prime, 5 prime to 3 prime, and then it added the wrong base. So how is it going to recognize that it's a wrong base to begin with? Remember, when you have base pairing, you always have a pyrimidine that pairs up with a purine. Pyrimidine is 1 ring, purine is 2 rings, so here's 1 ring, here's 2 rings, okay? So we do base pairing, base pairing, base pairing, and then you come to one point in which it makes a mistake and it puts 2 pyrimidines together. So polymerase is going to feel it that something is wrong with the size of the double helix, because it was going like, okay, double helix looks good. Polymerase doesn't see, so it's going by feeling. Right? So I'll go like closed eyes, no eyes in polymerase. It's going by feelings, like DNA looks good, DNA looks good. DNA looks skinny. That's a problem. I made a mistake. So it goes back, chops up a piece of that DNA by exonuclease activity, and then it starts again. DNA looks good, DNA looks good, no mistake. And now let's say it found 2 purines that got put together. DNA looks fat, DNA feels fat. I go back, I chop it off, I keep on going, and I add the right nucleotides, the right base pairing. Okay. So this is what you call a proofreading mechanism. Okay. Now we're going to see which enzymes are very good at proofreading. So looking here at your prokaryotic DNA polymerase 1, okay? So how is it good at making polymers? Look, it's going to do polymerization just like every other polymerase, 5 prime to 3 prime. And then this thing has 3 prime to 5 prime exonuclease activity. So it stopped and went backwards and then chopped off at the end of that polymer, right? So it has a nuclease, and then it kept on going. Now remember, DNA polymerase 1 is the guy important for removing those Okazaki fragments, right? So DNA polymerase 1, when it gets, to the end of removing those fragments and taking out the RNA and putting in the DNA, it actually checks a little bit ahead of its work because it can correct mistakes that were left behind and not noticed by itself, basically, or by DNA polymerase 3. So it also has 5 prime to 3 prime exonuclease activity. So it can correct going backwards, it can correct going forwards. So this is a very, very important proofreading ability of DNA polymerase 1. Okay, everybody got that? All right. All right, so here talking about DNA polymerization and how things go well with proofreading and correcting. And then here I put this slide, also coming from the book, showing that there are certain drugs that interfere with the action of polymerases, and there are certain drugs that pretend to be nucleotides and mess up polymerization because they stop the polymerization. Okay. So there are several mechanisms that you can explore to stop polymerization of certain species that you don't want to proliferate. For example, you can stop polymerization of certain viruses you can inhibit certain viral polymerases by using these types of drugs. Okay? So here I'll give you a couple of examples of what I meant by that. Okay? So you have certain antiviral drugs, and here I have the example of acyclovir. Now look at the name of this drug. Acyclo, it means it doesn't have a circle. Vir, it means it's working on viruses. Okay, so when you look at this drug, acyclovir, you look at the structure of acyclovir, look at this thing. It has guanosine right here. I mean, it has a guanine, purine right there. It doesn't have the sugar ring right there. So this thing is without the circle, without the sugar ring structure. Okay, but it has a leaving group right here. Okay, so what is happening here? But it doesn't have here, see, it doesn't have 3' OH, 5', it doesn't have any of that stuff here. So what happens here, this drug, when you put it in cells that are infected by certain viruses, especially herpes viruses, okay, you can stop the activity of the DNA polymerase of the virus, because this drug is going to bind to the viral DNA polymerase and is going to inactivate the action of that DNA polymerase. And is actually quite effective against chickenpox, that is herpes virus 3, and by that token also with shingles, that is also caused by the same herpes virus 3. Okay, so this is a type of drug that is very good at blocking DNA polymerases of those viruses. Those are DNA viruses. Now, one thing that should be coming up in your head is, can this just stop the host DNA polymerase as well, like eukaryotic DNA polymerase? Couldn't it just kill the host cell too? Well, it could, but it doesn't. Because this drug will bind strongly to the viral polymerase, like 100 times more, like 100 times stronger to the viral DNA polymerase than it would bind to the host cell. Okay, so it's actually selective for a certain type of viral polymerase. Okay, so there are certain drugs that you can use for that, for inhibiting viruses. So here I have a couple of slides that summarize all the issues of DNA polymerization and comparison with eukaryotic polymerization and how those cells solve the problem. So you can read that on your own. I really took a lot of time to put those summary slides in a logical order. Okay. Okay, and remember, this is your clamp. I like this picture of the clamp around the double helix of the DNA here that is looking from, instead of like this, is looking like this, right, from a different perspective. So here's the DNA floating inside of that positively charged beta ring, beta clamp. Okay. All right, so here is another summary on removing the primers. DNA, right? Replication new DNA. Then we're going to look at transcription. That's making RNA from DNA. Right, so that's different. Okay? Let's not get into technicalities of what primase is doing. All right, replication is making new DNA. That's it. So now let's see problems with DNA damage. How do you get DNA damage? So we're just going to start on how we get the damage, right? If you get damage, doesn't mean you're going to have a mutation. Damage means maybe you can undo the damage. When you do not correct the damage, when it becomes permanent, then you call it a mutation. All right, so this, you can read that on the description of the chapter. All right, so DNA damage. What is causing DNA damage? Okay. So look at this. Every second is more dramatic that you read this. Oh, DNA in each cell of your body is getting damaged. This is like close to Halloween, right? So chemical bonds are breaking. Some DNAs are strands are snapping. You have nucleotides flying off the DNA. Okay, look at this. Each cell loses more than 10,000 bases per day. Every day you are losing bases out of your DNA. Okay, those are your nitrogenous bases. They mainly are purines, could be some pyrimidines. You're losing it. It's falling off the DNA. Okay, that happens at regular body temperature. What's your body temperature? 37 degrees Celsius. So you don't have to boil the DNA to start losing bases. Your own body temperature, slow cooking throughout life. Okay? Now, if the damage is not corrected and you incorporate it when the cell divides, then you can have mutations. Okay? Exposure to certain chemicals, including smoke. We're not only talking about cigarette smoke, we're also talking about other forms of smoke, you know, like even medical marijuana, that's smoking, okay? It can cause DNA damage, right? Smoke from industry, and then you have sun, UV light can damage your DNA, radiation, chemicals, a lot of stuff can damage the DNA, okay? So when you do not correct the damage, this is when you get mutation. Mutation is a natural process that happens throughout evolution. It's actually what allows evolution to happen. If you do not have a certain level of mutation, there is no way of having evolution. Okay? So mutations are those permanent damages that became incorporated into the new DNA that was done. How is it that mutation happens? Well, it's not something that is predicted and you can actually direct the selectivity of the mutation to go on. It happens at random. Okay? Very bad mutations will kill the cell, you'll never see it again. Good mutations that actually improve some efficiency of some enzymes, they will be kept. So that will be passed down the line. So that will be considered natural selection. So natural selection comes from natural mutations. All right. You have different types of mutations depending on how you're looking at it. If you look at the chromosome, big pieces of chromosome, you call that chromosomal mutation. If you look at sequence of the nucleotides in a gene, you call that gene mutation. Okay, so you have certain terms that are related to big chunks or tiny pieces. Some mutations happen naturally, so you call those spontaneous. Some mutations happen because you introduced something to cause the mutation. So those are called induced types of mutations. Like when you put in a certain drug that is going to interfere with your DNA, then that's induced type of mutation. Now, this is important here. Somatic cells, those are all the cells in your body except your germ cells. So in somatic cells that are differentiated, when mutation happens, that stays with you. You are never going to pass it to your offspring. You're never going to pass it to your kids. If mutation happens in the germline, in your gametes, okay, eggs, sperm, if mutation happens there, that's a problem because that's what you can pass on to your offspring. Everybody got that? All right. So, possible causes of mutations. Here you have your spontaneous mutations. Okay? Those are their uncorrected errors and they happen naturally. What kind of spontaneous mutation? First thing that you're going to look at is at the purination. You see this purine right here? It can just fly off one strand of DNA naturally. Okay, so it gets it just flies off because you can have a water molecule that can make this bond here to undergo the hydrolysis. So this piece falls off, and now you have a site here in your double helix that is missing. Well, here's not showing the double helix, but you're going to have one site that is not going to have a purine. So you're going to call that site in your double helix, you call that a purinic site. It's missing a purine. All right, so this happens naturally. How often? Look at this. About 10,000 of those fly off your DNA in one day. You don't have to do anything about it. They'll just fly off on their own. Okay? All right. Other types of spontaneous damage. You can have deamination. So you think about an amino group. If you look at cytosine, you have an amino group right there. And you can have deamination. This amino group will fly away. And let's say it gets converted to another group that is a carboxyl group here. If that happens, you are changing this pyrimidine from being cytosine to being uracil. That is a problem because uracil should not be in the DNA to begin with. Okay? Uracil binds to what? U binds to? Is it A or T? T. Okay. U binds to T and C binds to G. So if you do a deamination here, you end up with this in the DNA. This thing is going to bind to not what it's supposed to bind to. Okay, it's not going to form 3 hydrogen bonds. It's going to form 2 hydrogen bonds, and then your DNA is screwed. All right. Let's see what happens here with adenine. If you lose the amino group, it becomes this other thing here that you don't even see in the DNA at all. Okay, hypoxanthine, it shouldn't be in the DNA molecule. So that is a problem. You're not going to have proper hydrogen bonds. So this is when I put this comment here saying that deamination of here methylcytosine can give you thymine. So happens. That is C going to T, right? And it happens in several aspects of life, including in your DNA, right? So Sh2 happens. What are problems that, besides deamination, that you could have? Okay, just this one last slide here. You could have isomers that could be shifting from one form to another. You call those tautomers. So you can have tautomeric shift from something that is a ketone to something that is an enol. Okay? When tautomeric shift happens, your DNA is going to be messed up. All right, so I'm going to stop here. And we're going to pick up from there.